And thank you for the organizers for having me here to share some thoughts with you. Today, rather than talking specifically about modeling neuroscience itself, what I'm going to do is talk about how some neuroscience ideas can extend beyond their usual substrate. And we're interested in collaborations and hopefully some work that can shed light on the origin and also some fundamental principles that are useful to neuroscientists. And all of the data sets, the software, all the primary materials are all here. And here are some personal thoughts on what some of our work means. So here is the summary of what I'm going to talk about today. And that is that one of the things I'm most interested about in neuroscience is that it's uniquely poised to study multi-scale intelligence. And I'm going to make the claim that the tools and concepts of neuroscience are applicable far beyond neurons, in particular through the evolutionary conservation of both mechanisms and strategies that are used. I'm going to talk about bioelectricity as a specific example of cognitive glue that holds together various emergent collective intelligences. And I'm going to describe morphogenesis as a model system for studying unconventional cognition. And I'm going to talk about the implications at the end for evolution, bioengineering, regenerative medicine, and maybe even consciousness studies. What I'm really interested in my group is to create a framework that will allow us to create, recognize, and relate to really diverse kinds of agents, regardless of what they're made of or how they got here. So all of these kinds of things, familiar creatures such as primates, colonial organisms, swarms, but also organs and tissues, synthetic biology, engineered new life forms, and also AI, whether software or robotically embodied, and potentially even exobiological agents someday. And so what I'm interested in is frameworks that move the experimental work forward, that give us new capabilities, new biomedicine, and some new ideas about ethics of how to relate to beings that are not exactly like us. And this is, of course, I'm not the first person to try for something like this. Here's Wiener, Bigelow, and Rosenbluth back in the 40s trying to show what are the transitions that can lead from passive matter all the way up to complex human metacognition and so on. And one particular interesting thing is that I often wonder, when some of the founding figures of trying for this kind of understanding of what's going on in the mind and in cognition, what would have happened if the biologists had come to them and said, actually, we made a mistake. It's not really in the brain. It's in the liver, or it's somewhere else. Would they have said, no problem, our models still work, it doesn't matter? Or would they have said, no, you must be wrong. Our models say this is exactly why it's in the brain. And so we're interested in this question of, well, what exactly, if anything, is special about the brain and the nervous system? And I find it very interesting that Turing, who was, of course, extremely interested in generic problem-solving machines and, in general, intelligence through plasticity and reprogrammability and very diverse embodiments, also became interested in morphogenesis. So he wrote this very famous paper on how the chemicals in an early embryo actually arise in terms of patterns, right? How you get chemical patterns. And so you might wonder, why would he be interested in this kind of thing? And I think because he saw very clearly that the self-assembly of bodies and the self-construction of minds are very isomorphic problems. I think there's a deep symmetry in these kinds of things, and that's what I'm going to talk about today. Is what I think are very common features of understanding cognition and understanding developmental biology. So we'll talk about three things today. First, some comments on the evolutionary origin of neural mechanisms, and also we'll talk about in detail morphogenesis and then the implications. And one interesting thing about neuroscience is that you can, and this is a useful exercise that I used to have my students do, where you can take a neuroscience paper and you can do a find replace. So anytime it says neuron, you replace the word cell. If it says milliseconds, that becomes minutes. And instead of movement and behavior in three-dimensional space, we analyze something called morphospace, anatomical morphospace. What you get then is a very readable developmental biology paper. It's an interesting exercise, and I used to have students do this by hand, but now we have this very simple kind of AI tool that does it for you, so you can go here and play with it, and this is the paper that's associated with it. You can put in a neuroscience abstract and see what the corresponding developmental biology looks like. And there are some interesting experiments that show the interconnectivity of the memory of behavior, functional behavioral memories, and the memory of morphogenesis in the body. So for example, these are planaria, these flatworms that you can chop into little pieces. We'll talk a lot more about them later in the talk. But basically what happens is that each of these pieces regrows a perfect little worm. Okay, so every piece knows how to make a perfect little worm. But remarkably, if you train these animals on a particular task, such as place conditioning to feed on these bumpy surfaces, and then you amputate their head, which contains a centralized brain. The tail sits there doing nothing for about 8 days. There's no behavior without the brain. Eventually, they grow a new brain here. And when they do, you can recover the original information. You do behavioral testing, and you find out that they actually still remember the information. So what you have here is not only the storage of information outside the brain, but actually the ability to imprint that information onto a brand new brain that forms after regeneration. And this may have implications for human medicine, let's say replacement of degenerative diseased tissues with naive stem cells and so on. Of course, we don't know how this is going to work out in humans, but the ability of information to move through the body, and this kind of example that shows you the consilience of behavioral memories with anatomical memories already tells us that there's an interesting relationship here. So this is the journey that we all take. We start off life as an unfertilized quiescent oocyte, a little blob of physics and chemistry, and then very slowly, very gradually, we become one of these things, or even something like this. And the important thing about developmental biology is that it offers no place to draw a bright line that says, well, before this was a system of physics, but now you have mind, and now you are amenable to behavior science and psychoanalysis and so on. There is no specific place where that happens. It's all very, very slow and gradual. And what you have here is a dynamically self-modifying computational medium that builds itself and scales up its various cognitive powers. And we need to understand how this works. We need to understand the scaling of the competencies from here to here as a story of transformation. Interestingly enough, the way these embryos appear is already a dynamic process. So when you're looking at a blastoderm and you say, oh, there's one embryo, what are you actually counting? What is there one of? There may be 100,000 cells. What is there one of? Well, what there's one of is alignment, both physical and behavioral. On the part of these cells, they are all making the same journey in anatomical space. They are all committed to building exactly this thing. And if you make some scratches in this blastoderm, each of these little islands, before they all heal up and become one contiguous blastoderm, each one of them will self-organize their own embryo, and then eventually you get conjoined twins and triplets and so on. So the number of selves, the number of cognitive individuals that can be formed out of this material is anywhere from 0 to half a dozen or more. And unlike with computer memory, we actually don't have a good way of formalizing what is the carrying capacity of this medium. How many individuals can be formed here? But clearly it's more than one. And this already has interesting parallels with the same kind of thing in cognitive science, where you have split brain patients and dissociative identity disorders and so on, when there are multiple individuals running on the same hardware. So we're very interested in this question of self-organization and scaling, and where both minds and bodies come from. Now, one important aspect is that intelligence, as many people study it, takes place in the three-dimensional space of behavior, so motility. So medium-sized objects moving at medium speeds through the physical world, we're pretty good at recognizing these things as intelligent behaviors. But biology does this in many other spaces. The perception action loop can happen in many other kinds of problem spaces. So the space of transcription, so all possible gene expression states, physiological state space, and the one we're going to talk about mostly today, which is anatomical morphospace. And so living systems navigate all of these things, not just three-dimensional space. And we need to remember that when we talk about, for example, embodiment. Lots of people have made the claims that, for example, neural organoids or brain organoids in culture are not embodied and thus lacking, certain aspects of cognition. And we need to remember that this visual space of motility is not the only space. They are, in fact, navigating these other spaces. And I think actually what evolution has been doing is pivoting some of the same tricks in terms of navigating spaces all the way from metabolic, physiological, transcriptional, anatomical, and eventually, when nerve and muscle came on the scene, behavioral, and then linguistic, and who knows what else. But all of these spaces are ones that biology navigates using some of the same strategies. And the interesting thing about how it does it is that it's actually a very deep multi-scale competency architecture. So nothing is really centralized. So inside the brain, so there's the pineal gland that Descartes really liked because it's a unitary organ. But inside of that, if he had had good microscopy, he would have seen that there isn't one of anything. It's actually made of cells, and inside each of those cells is all of this stuff. Okay, so we are all, not just the bees and the ant colonies and the termites and so on, all of us are a kind of collective intelligence. We are all made of parts. This is the kind of thing we're made of. This is an organism called a lacrimaria, and it's a single cell. There's no brain, there's no nervous system. You can see that it's got significant competency in its own very small kind of cognitive light cone that it has here. And the thing about the material that we're made of is that it's not just nested scales of organization, it's not just complexity. The material has agendas and competencies at every level. And so, for example, even this little guy is made of molecular networks, and it turns out that molecular networks can exhibit 6 different kinds of learning on their own. You don't need the cell, you don't need a nucleus, just the gene regulatory networks itself can do learning, including Pavlovian conditioning. And we're in the process of developing apparatus that actually exploits that to train cells and do drug conditioning and other things that are interesting for biomedicine. But even below the cellular level, you already have capacity for learning and decision making and so on. So what we have here is this architecture that at every level is able to solve different kinds of problems and exert different kinds of competencies to navigate that space. But one thing that's very important about such an architecture is you need something that we call cognitive glue. You need something that binds these things together towards the emergence of a larger scale system that has goals and preferences and memories that don't belong to any of its parts. So when a rat is trained to press the lever and get a reward, the cells at the bottom of the feet interact with the lever, the cells in the gut get the delicious reward. No individual cell has both experiences, but it's this rat, it's this collective that has the associative memory. And so we know what binds these things together in the behavioral space. And so here is the hardware, and it's the physical neural networks, and the physiological kinds of computations and other things that progress on these networks. And what they do is typically they issue outputs to your muscles to move you through three-dimensional space. And then we try to do processes such as neural decoding to try to read these physiological states and extract semantic content. But what they're doing is they're moving you through three-dimensional space. And one thing that's interesting about this is that this is actually an extremely ancient architecture. This long predates the evolution of neurons. This idea that cells make electrical networks that then have a very particular kind of computational dynamics that runs on them. What it was doing before, what did these networks think about before they were thinking about moving a body through three-dimensional space? Well, they were thinking about moving the configuration of the body through anatomical morphospace. These electrical networks were issuing commands to all cells, not just muscles, but all cells, to move the configuration of the body through the space of possible anatomical configurations. And what we're doing in our lab is developing a very parallel kind of effort to neural decoding to try to extract the semantics from these non-neural electrophysiological events to understand how the developmental decision-making happens. And so now what we'll do is we'll talk about some examples of what happens during morphogenesis as a model of collective intelligence. So it's carried out by groups of cells, and I'm going to make the claim that it not only exhibits a kind of collective intelligence, but also that, much like in the brain, what holds it together and allows it to do things that the individual parts don't do is electrical networking and the information processing that it does. And when I say intelligence, I mean William James's definition. Obviously, it doesn't encompass everything. There are many definitions, but William James said intelligence is the ability to reach the same ends by different means. And I like that a lot. It's a very cybernetic definition. It doesn't say anything about brains or scale or in fact, the problem space that you're working in. Just the ability that you have some degree of competency to reach a goal in your problem space, even when things change. So let's take a look at what development does. First of all, development is a process that can go from a starting condition such as this egg to a species-specific target morphology like this. And for the first time, we encounter something very interesting, which is that that process is incredibly reliable. So most of the time, this goes off without a hitch. It's an amazing process that does this reliably. It does occasionally make mistakes. And so for the first time, we encounter this notion because chemistry doesn't make mistakes. Chemistry just does what it does. But developmental biology and cognitive systems can make mistakes. And they can make mistakes when they take actions that block them from the goal that they're trying to reach. But what's important is that the reliability or the emergent complexity that happens here, obviously there's a huge increase in complexity here, these are not why I'm calling it intelligence, okay? And a lot of people have this picture that this is a rote mechanical process. It's just sort of the workings out of chemical rules, and then it's kind of an open loop emergent process. Eventually you get complexity. That in fact is not what it is. I'm going to show you some examples which explain why I think this is a problem-solving process. It's not just the complexity, and it's not just the reliability. The first thing we know is that if we chop some of these early embryos into pieces, including human, what you get is not half bodies. You get monozygotic twins and triplets and so on. So the system very rapidly realizes that half of it is missing, and it restores the rest. And so if we boil down all of anatomical space and into just two axes, you see that it's able to start in different starting positions and get to the ensemble of goal states that represents a normal human morphology, even when you're starting it off in different positions, and it can avoid all kinds of local maxima and so on. So that's interesting. It can start off at different positions. What it can also do, and this is a process known as regeneration. So some animals, like this axolotl, can regenerate body parts. So these guys can regenerate their limbs, their eyes, their jaws, their ovaries, lots of different organs. And so what happens here is that when this is amputated, this limb is amputated at any particular region, the cells will grow exactly what they need, and then they stop. And that's the most amazing thing about regeneration, is that it knows when to stop. When does it stop? When the correct salamander limb has been completed. So this is a kind of anatomical homeostasis. You can think of it as a means-ends analysis, that when there's a delta here, it will try to reduce that error as much as it can, and then it stops. There are also some amazing examples of the fact that this happens often via different paths. I don't have time to show you that now, but basically it doesn't always recapitulate the same path that it used to grow during development, but it ends up in the same place. And so what we can see here is that it's not simply some kind of a feed-forward emergent process. It's actually a closed-loop error minimization scheme. And for example, you can see that here. This is something we discovered a few years ago when these tadpoles, here are the eyes, the brain, the nostrils, the mouth, when these tadpoles need to become frogs, all of these organs have to rearrange themselves in order to become a normal frog. And it turns out that, counter to the initial expectation that this is somehow hardwired and you just move the organs the right amount in the right direction and you get your frog, we made these so-called Picasso tadpoles where everything is scrambled. So the eye's on top of the head, the mouth is off to the side. These guys become quite normal frogs because all of the organs will move in novel paths to get to where they need to go. until you get a normal frog, and then they stop. In fact, sometimes they go too far and actually have to double back. So what the genetics gives us here, and we'll talk more about this at the end, is not some kind of set of hardwired rearrangements. It actually specifies a system that executes a very flexible program of moving things around until they get to the correct place. And now the obvious question arises, or one of the several questions arises, is how does it know what's correct? Okay, where is the encoding of the set point for this homeostatic process? Where are they going, all of these cells in their journey in anatomical morphospace? So what we've been studying is the idea that very much the way that nervous systems can encode specific goal states as memories, it turns out that something like this happens in all of your body tissues. So these ion channels, these electrical synapses, and the neurotransmitters that regulate some of the stuff that happen in neurons actually operate in every cell in your body. Every cell of your body has ion channels. Most of them have these gap junctions with their neighbors. And evolution discovered back around the time of bacterial biofilms that these electrical networks are an amazingly effective way of storing information and integrating it across space and time. And so just as neuroscientists study the meaning of the physiological signals in the brain, we do the same thing. This is an early frog embryo. Okay, so the mechanisms, the molecular mechanisms that neurons use are in fact very ancient. They're conserved all the way to pre-multicellularity, and one of the things that they do have that's different is speed. So here we're talking millisecond kinds of events, and here minutes or hours. Now we've developed some tools to read the electrical state of non-neural cells. So you're seeing here some cells explanted in culture. Here's a frog embryo. All of this is using, in this case, a voltage-sensitive fluorescent dye. We also have some genetically encoded reporters, such as neuroscientists use in optogenetic studies and so on. But we can track all these electrical kinds of conversations that these cells are having with each other. We do a lot of simulation all the way down to the molecular networks that express the different channels, but then the collective behavior of the bioelectric gradients, right? And try to actually understand how they do pattern completion and what are the attractors in these spaces and so on. And so we have these models and we analyze them in these ways. And I want to show you a couple of examples of what non-neural bioelectricity actually looks like. So this is a time-lapse movie of an early frog embryo putting its face together, and the grayscale levels indicate bioelectric state. And what you can see here, this is one frame out of that movie, is that prior to the formation of the craniofacial structures, in fact, prior to the necessary genes turning on and off, already this tissue has a layout of what it's going to build in the future. So here's where the animal's right eye is going to be. Here's where the mouth is going to be. Here are the placodes. And as I'm going to show you in a minute, this pattern is instructive. It actually tells the cells where to put the different structures. So we are reading out from this tissue the plan that it has for building it. I mean, it's literally a pre-encoded set point. This is not just the output of some kind of cellular automata or something that just sort of emerges over time. This is a pre-existing map of what the system is trying to reach. And it will try to reach this state even if you try to block it, as we talked about. So here, this is a different kind of imaging. Again, you can see there's a few neurons here, but everything else, all these other cells are non-neural cells, and there are multiple scales, both spatially and temporally, by which these different electrical systems interact. And it's not just, this bioelectricity isn't just the cognitive glue that binds individual cells towards common purpose within an embryo. It actually operates across groups of embryos. So you can go beyond the organism scale, and you can see here, if I poke this animal, within about 30 seconds, these two find out about it, and there's water in between, so it propagates across space. There's a signal that involves calcium and ATP and some other things that allows, and you can see here waves, these injury waves propagating across groups of animals. And in fact, it turns out that groups of embryos together are much better able to solve various teratogenic challenges than individuals. So even there's a new level of collective intelligence where they can sort of solve problems as a group better than they can as individuals. So one of the ways that this collectivity arises is through the scaling of the set points that these systems can manage. So you can think about the size of the largest goal that they can maintain, which I think defines their cognitive light cone. So individual cells have little tiny goals all around maintaining physiological and metabolic states on this tiny scale. But what evolution and development does is wire them together with electrochemical networks that maintain grandiose goals, huge goals like building a limb here. And we know this because if you perturb them, they will come back. They will try to reach this despite different perturbations. Now, no individual cell knows what a finger is or how many fingers you're supposed to have or anything like that, but the collective absolutely knows, and we know that experimentally it will try to come back to this location. Actually, if you do this 5 or 6 times, it eventually gives up. So it has the ability to detect. Eventually, it has this like metacognitive thing where it will just stop trying. But for the first N attempts, it will do a very nice job of rebuilding. But that ability to join into larger networks that can store large and remember larger goal states has a failure mode, and that failure mode is cancer. So what can happen is that when individual cells disconnect from that network, they can no longer remember this gigantic goal state they were working on. They basically roll back to their ancient unicellular past. This is human glioblastoma. Basically, we can study this reversion to cancer as a kind of dissociative identity disorder of the collective morphogenetic intelligence. It literally fragments into pieces with very tiny goals. And once you start thinking about this as cancer not being more selfish, the way that it's sometimes modeled in the game theory models and so on, it just has smaller selves. The boundary between self and world has shrunk for each of these cells. That leads directly to a kind of therapeutics. You can imagine targeting that mechanism and actually doing a kind of forced integration where so here's a tadpole that's been injected with a human oncogene. Already you can see immediately the voltage is off. These cells depolarize. They're going to start crawling away and metastasizing. As far as they're concerned, the rest of the body is just environment, right? The border between self and world has really shrunk to the single cell level, and they don't care about the rest of the body anymore. They're just maintaining their own tiny little goals. But what happens is that if along with this human oncogene, you inject a specific ion channel RNA, which forces the cells to acquire an electric state that keeps them in good connection with the rest, then here you will see this is the same animal. There is no tumor. The oncoprotein is blazingly expressed. It's labeled with red fluorescence. In fact, it's all over the place, but there's no tumor because the genetics isn't what drives. What drives is the decision-making of the collective, and in this case, these cells have been reattached and forced to remain part of this electrical network, and they go on building nice skin and muscle and everything else, despite the fact that their genetics have these really nasty oncogenes. Okay, so now how do we do this, right? What are the techniques by which we can read and write new information into the electrical networks of these cells? Basically, we don't do any, there are no applied fields, magnets, waves, frequencies, no electromagnetic radiation. We take everything from neuroscience. We do exactly what neuroscientists do, which is we can change the topology of the network by targeting these electrical synapses, so we can alter the synaptic plasticity across these cells, and we can target the actual electrical state of these cells by modifying these channels. You can modify them genetically. You can use ion channel targeting drugs. We use optogenetics and light to turn these things on and off. So what we do is we basically are hacking the communication interface that cells normally use to control each other's behavior. Right? The only reason these things I'm going to show you in a minute work is because that is what the cell collective is already doing. Okay, this is whereas before we were sort of trying to read the mind of this network. Now we're incepting new memories and new behavior patterns in here, just like people try to do in the brain. So what happens when you try to do this? Okay, what can you actually do with this? So this is one of I could show you many things, but this is one of my favorite examples. This is an early frog embryo, and if you remember that electric face pattern that I showed, there was one particular little eye spot that's where the eye is going to come out, and we wondered what would happen if we recapitulate that eye spot somewhere else. Okay, and so if we inject some RNA encoding particular kinds of potassium channels that will set the cell voltage to roughly that kind of spot, and we hit cells that are normally nowhere near the head, such as these gut cells, turns out that they will make an eye. Okay, and inside the eye is lens, retina, all this other stuff, exactly as you would expect. Now, that teaches us a few things. First of all, that the bioelectric pattern is instructive. This is not just about making defects, it's about telling groups of cells what organs to build, so it's actually functionally instructive. It's extremely modular. We didn't tell these cells how to build an eye. We have no idea how to build an eye, and we certainly aren't able to manipulate all the different stem cells and the different spatial relationships between them and all the genes that have to be turned on and off. We provided a very low information content, high level subroutine call, which says build an eye here, and that is what the cells did. So we're now in the process of discovering more of these signals that cause specific large scale changes. Interestingly enough, the developmental biology textbook said that only the tissue up here, only the anterior neurectoderm, was competent to build an eye. And that's true if you use the so-called master eye gene called PAC6, which is what normally is sufficient for making ectopic eyes. PAC6 doesn't work anywhere roughly behind this location. But the bioelectric prompt works pretty much anywhere in the body. And so that reminds us that when we see limitations of competency on some particular system, that may well be a limitation on our competency to provide the right prompts. Okay, that may not be a competency of the system itself. And the other cool thing about this is that this is a section of a lens sitting out on the flank of a tadpole somewhere. If you only inject a few cells, so here these blue cells have been injected, there's not enough of them to make a proper eye, but they can tell, and what they do is recruit a bunch of their neighbors here. These are not blue, these are cells we never touched. So we told these cells make an eye. They then recruited a bunch of their neighbors and said, you now have to participate with us and make this eye. So all of this works, this self-scaling, and by the way, there are of course other collective intelligences that scale. The ants and termites will recruit their nestmates to move large objects and so on. So all of these competencies, this ability to use top-down, very simple top-down controls to rely on the self-scaling and the competency of organogenesis, is specifically because all of these systems are already primed to accomplish specific tasks when given specific signals. They do not need to be micromanaged. Here's another example, which is what we're doing for regeneration. So frogs, unlike salamanders, do not regenerate their legs in these adult stages. So we wanted to know what kind of signal could we give them to allow them to do this. And what happens is that we made a cocktail of ion channel and other drugs that basically kickstart a regeneration. And within 45 days, you've got some toes, you've got a toenail, and eventually a pretty good regenerating leg. Now, in the latest experiments, 24 hours of stimulation gives rise to a year and a half of leg growth. And during that time, we don't touch it at all. And that's because, just like we're familiar in behavior science, when you communicate with another person or with an animal, you're providing signals. It is not on you to rearrange all the synapses in their material to make that information sink in. The system itself does all of that. You are providing high level signals. The system is primed to take all of that and make the molecular changes needed for them to become memories, to become executed goals, and so on. And exactly the same thing is here. And we are currently trying to move, so that was, we figured that out in frog. Now the next step is mammals, so we're trying to do this in rodents with wearable bioreactors in collaboration with Dave Kaplan. And here I have to do a disclosure because there's a company called Morphoceuticals Inc., whose job it is to try to figure out how to induce things like limb and other organ regeneration with top-level stimuli, with convincing the cells to go to the leg building route, not the scarring route, in ways that are not micromanaging, not scaffolds, not trying to convert stem cells or anything like that. Okay, so the next thing I want to show you is something interesting about memory in these systems. So again, here are our planaria. These planaria are remarkable. They are incredibly regenerative with almost 100% fidelity. They are also cancer resistant. They are immortal, so they don't. These asexual strains don't seem to age, and they're also incredibly resistant to transgenesis. And if we have time, I'll talk about this at the end. They have an incredibly noisy genome, and I actually think that's why they have these amazing properties. It's kind of counterintuitive. We'll talk about that. And so one thing you might ask is, how does each of these pieces, right? So you cut a little piece. The record is, by the way, something like 276 pieces. You cut a little piece here. How does this piece know how many heads it's supposed to have? Okay, And we studied this question, and we realized that they have this bioelectrical pattern that we can read out that says one head, one tail. And when you cut this, you then have 100% of the time you have a normal animal with one head, one tail. But what we were able to do using drugs that target this bioelectrical pattern is induce a system that now has this kind of pattern at both ends. And when you cut that animal, you end up with an animal with two heads. And this is not AI, it's not Photoshop. These are real two-headed worms. And that's because this bioelectrical pattern is the memory. It is the encoded representation of what a planarian is supposed to look like. And once you've changed it, the cells will do whatever that pattern says. Really critical to note, this electrical pattern, this memory is not a readout of this two-headed animal. This is a readout of this perfectly normal one-headed animal where the molecular markers are anterior in the head, not in the tail. The only thing wrong with this animal is that it has a different representation of what it will do if it gets injured in the future. And I think this is the beginnings of a very simple version of the kind of time travel that we can do with brains, where we can imagine scenarios that are not true right now. That's what's happening here. This is a counterfactual memory. This is not true right now. This has two heads. This animal does not have two heads right now, but that is what it will do if it gets injured. If not, it stays a latent memory. So the body of a planarian can store at least two, and I'm sure it's more, but these are the two we've nailed down so far, at least two different representations of what the goal is. Where is it going in anatomical morphospace is encoded in this electrical network, and it's labile. Now, one reason I keep calling it a memory is because if you take these two-headed worms and you chop off, again, the primary head, you get rid of the ectopic secondary head, and you have this middle fragment, it will again give rise to two-headed worms with no more manipulation. It's permanent. So this system has all the properties of memory. It is long-term stable. It's rewritable. It is not entirely determined by the genetics. So different memories can sit and run on the same hardware. So the question of what determines how many heads a planarian has, is it the genome? It's a subtle question. The genome determines the initial state, which reliably will say one head, but that is not where the information actually sits, and you can rewrite it. And here you can see these animals in behavior, and in fact we can set it back now. We can rewrite that pattern back to one-headed, and then these guys will return. There's something else that's interesting that we can do with these. You can make this strain of two-headed worms. You can make another one which we call cryptic worms. And the thing about cryptic worms is they're confused in that when you cut fragments, so let's say this fragment up here and this fragment down here, some of them will form two heads, some of them will form one. It's a stochastic decision now. So what you've done here is destabilize that pattern, and with Gio Pizzullo, we analyzed this thing as a kind of perceptual bistability. The bioelectric pattern is poised in a way that it can be interpreted in multiple ways, such as Necker cubes and this rabbit-duck illusion, that kind of thing, where the system that reads the electrical pattern stochastically at a rate, at a frequency of, you can make a transition table, it's roughly 3/4 to 1/4, where it will interpret that pattern as either two heads or one. And so we've been making models of these kinds of things to try to go from the bioelectric circuit, which we now understand to some extent, to the system level behavior to try to understand how it recovers specific patterns when things are missing, and in fact how it makes mistakes. One thing that in keeping with the idea that much like with learning, is that the same hardware can have lots of different memories. What we can do is take a planarian with a nice triangular head here, and by perturbing that bioelectrical network during regeneration, we can get them to make flat heads like a P. felina, round heads like an S. mediterranea. So no genetic change here. They're perfectly happy that that exact same stock hardware is happy enough to visit the attractors that normally are occupied by other species of planaria, about 100 to 150 million years of evolutionary distance. Okay, but if the pattern memory says that this is the kind of head that you should have, then that's what it builds. And by the way, not just the shape of the head, but the shape of the brain and the distribution of stem cells becomes just like these other species. Okay, so. what we've talked about here is morphogenesis as a model of collective intelligence, which solves a number of problems in reaching its goals in the anatomical space. We've shown how we can use some tools and concepts from neuroscience to address this, and so we've done. There's lots more that I can't show you now, but we've done things with memory blockers and with hallucinogens and with various anxiolytics and SSRIs, and all of these map very well onto this idea of a system making decisions and navigating that anatomical space. And once you do that, you unlock some biomedical applications that are not readily available from the standard bottom-up kind of view of the genetics as a simple mechanism that drives the outcomes. So in the last few minutes, I want to talk about some of the implications of all this. Here's a basic kind of observation. You take this tadpole, and what we've done here is, so here are the nostrils, the mouth, the gut, the brain. What you'll notice is that we've prevented the eyes from forming in the head where they normally form, but we've made an eye form on their tail. So the first thing is that eye precursor cells are perfectly happy to build an eye on the tail. It makes an optic nerve. The optic nerve does not go to the brain. Sometimes it synapses on the spinal cord here, sometimes on the gut, sometimes nowhere at all. And the key thing about these animals is they can see. How do we know? Because we built this automated system that trains them on visual tasks, and it turns out they can see quite well. Now, this is somewhat remarkable. We've made a system with a radically different sensory motor architecture. The eye doesn't connect to the brain. How is it that it works out of the box? Why don't we need some sort of additional rounds of evolutionary adaptation, mutation, selection of different circuits and so on? Why does this work on the fly? And I'm going to argue that the reason this works is because fundamentally all of morphogenesis, even normal morphogenesis, is a problem-solving in real-time system, that it never actually assumed the eyes were going to be there in the first place. And to give you an idea of what I mean is a few more examples. This is, because development is so reliable, we're used to thinking that it's a hardwired process. Acorns always give rise to oak leaves, and this is the shape, and it happens so reliably, so consistently, that we tend to think that this is what the genome actually encodes. It encodes this, and the conventional kind of approach is that the genotype encodes the phenotype. And what I'm arguing, and all the details are here in this paper, what I'm arguing is that there's actually a very important layer in the middle, which is this morphogenetic layer that I've been describing, which has a bunch of problem-solving competencies that makes a huge difference to evolution. The fact that this layer here from genotype to phenotype is not just complex, it's not just there's not just redundancy and degeneracy and pleiotropy and all of these things. It's not just complexity. It's the fact that the interpretation of the genetic information by these electrical, mechanical, and chemical networks, that is what gives rise to the anatomy. I've shown you this in the case of the two-headed frogs that the two-headed worms that override the standard genetics, the lack of tumor despite the presence of oncogenes, many examples where it is really the competency of this layer that makes an enormous difference. And so when you think about this map, we would assume that that's all that the oak genome knows how to do, is make this thing, except that along comes a bioengineer. Now it's a non-human bioengineer. This is a little wasp parasite. And what the wasp does is provide some chemical prompts to the cells, and then they end up building something like this. These are called galls, and you see something like this or something like this. We would have absolutely no idea that these cells, which normally make a nice flat green structure of a particular shape and size, that they were even capable of doing something like this if we didn't see this organism doing that. And so we now understand that reliable, yes, default behavior, yes, but actually the space of possibilities is vast, and this wasp is not building this the way that it would build its nest, which is like a 3D printer laying down glue and little pieces of material and meticulously building it by hand. All it did was drop off some signaling molecules and left. And that's because it can rely on the material's competency to interpret those signals and build the thing that it wants to build. It does not need to micromanage this process any more than we needed to micromanage the creation of the eye or the frog leg or anything else. So here I want to show you one of my favorite examples of actual creative problem solving in morphospace. Now, this is a cross-section through the kidney tubule in a newt. And what you can see here is that there's about 8 to 10 cells that work together to build this structure with an empty lumen in the middle. Now, one thing you can do with these cells is produce newts that are polyploid. They have multiple times the amount of chromosomes that they're supposed to have. And so instead of 2N newts, you can make 4N, 5N, 6N, and so on. And when you do this, something interesting happens. First of all, embryogenesis works. You still get a newt, which is already interesting. Then you find out that the cells actually scale to the amount of genetic material, so you get bigger cells. But the newt itself is the same size. And so how does that work? Well, that's because the cells automatically reduce the number of them that are being used to create this tubule. So that's the second amazing thing, is that the cells actually scale the number given their new size. And kind of the wildest thing of all is that when you make really gigantic cells this way, then one single cell will wrap around itself and leave a space in the middle for this lumen. Now, the reason that's amazing is that this is a different molecular mechanism. This is some sort of cell-to-cell communication. This is cytoskeletal bending. So what you have here is a kind of top-down control where in the service of this large-scale anatomical structure, the cells will use different molecular mechanisms. So they are using the tools that they have, the genetically specified affordances that they have, in new ways to get their job accomplished. So think about what this means. If you're a newt coming into this world, not only can you not count on what the outside world is going to be like, but you can't even count on your own parts. You don't know how many copies of your genome you're going to have. You don't know the size or the number of your cells. You need to adapt and use the mechanisms that you have to achieve the path through anatomical morphospace that you need to take. And so I think that what evolution produces is not solutions to specific problems, rather it produces problem-solving agents. And the idea is that at least nowadays, all of life is this problem-solving kind of architecture that doesn't take much for granted, and it even under standard embryogenesis, it basically is problem-solving. It is not assuming from the beginning, at least for most organisms. There may be a few exceptions, but for most organisms, I think it's not assuming what is going to what's going to happen. It does its best, and this is why the tadpoles with eyes on their tails works, and why the galls that are on these oak leaves work, and why we can make induced eyes with voltage signals, because all instances of morphogenesis is creative problem solving. It is the layer between the genetics, between the hardware produced by the genetics and the actual outcome, and that middle layer is doing the best it can to do something sensible in the middle. And all of that is explored in these kind of papers here. So this has really interesting evolutionary implications. In particular, it has what I think is a kind of intelligence ratchet that gets intelligence in the material of life going long before there are brains or neurons or anything like that. Basically, what happens is that because the laying position between the genetics and the phenotype is itself kind of a problem-solving system, that ends up hiding information from selection. And what I mean by that is think back to the frog face that was able to rearrange itself to get to the right configuration. When selection encounters an animal with a pretty good face of a frog, it doesn't know whether that's because the initial genetics were great and that's how it started, or if whether there was actually some structural problems, but the competency took over and it fixed it. The ability of the material to meet the same and despite different starting conditions hides that fact from selection. So we've modeled this. We have a couple of papers where we computationally model how evolution works on passive materials, which basically just do what the genetics say, and this kind of developmental competency where it actually tries to solve the problems of different starting positions. And what happens is that when evolution finds it hard to see the genomes directly, all of the work ends up being done on the competency property itself. So not the structural genome, but the competency of the material ends up being really cranked by evolutionary progress. But this becomes a positive feedback loop, because the more you increase the ability of the material to get to a correct state while the hardware itself is unreliable, the less the selection is able to see the quality of the genome. So progressively it becomes harder and harder to select for the best genomes, but what it can do is select for the most competent individuals, and that's why you get something like planaria, which are incredibly resistant to all sorts of things, to injury, to cancer, to aging, to transgenesis, and I think in those kind of animals that that spiral went all the way to the right, where basically a lot of the pressure was taken off of the genome and put into developing an algorithm that can make a proper worm no matter what happens to the genetics. It's very important that it does that because planaria reproduce by fissioning and then regenerating. So unlike the rest of us, when we get a mutation in a cell in our body, our children don't inherit these somatic mutations. But in planaria, that's not true because they split and then regenerate. Any mutation that doesn't kill the cell is propagated into the new generation, and in fact they're mixoploid. They're a total mess genetically. All the cells can have different numbers of chromosomes and so on, and so I think biology leans into this fact that the hardware is ultimately unreliable, and a lot of work goes into a creative problem-solving algorithm that gets the job done even when that happens. And so that ends up driving this kind of this kind of ratchet where the intelligence just keeps going up in the various spaces and then projected into different kinds of problem space long before we have nerve and muscle. And you can see something like this in this example of metamorphosis, that what it's doing is because of that creative problem solving that was already there at the very initial stages, when you have very simple creatures that are trying to meet specific anatomical, physiological goals and so on. By the time you get to memories and behavior, the material is really good at having optimization for the salience of the information, not for the fidelity of it. That is, not taking the information from the past too seriously on an evolutionary scale. This means the genetics of your lineage, and on the individual scale, it means whatever materials you've been given. What it tries to do is to remap it onto its new situation. So here's an example of this. You've got these caterpillars, soft-bodied creature that likes to eat leaves, and you can train it to find the leaves by crawling to a particular location on a particular color disc. Well, when these caterpillars metamorphose into butterflies, they have to build a completely new brain to now drive a hard-bodied creature that actually flies. And in order to do that, it basically dissolves most of the brain. Most of the cells are killed off, all the connections are broken, and then it builds a brand new brain suitable for this kind of lifestyle. But what has been shown is that these kind of trained memories actually persist, and so it's amazing enough to think, how is it that memories are persisting through the basically complete refactoring of the medium, of the cognitive medium. But more importantly, you couldn't even do this if you just try to keep the memories exactly as they were, because the precise memories of the caterpillar are of no use to the butterfly whatsoever. It doesn't eat the same stuff, it doesn't move the same way. What you have to do is take the deep lessons that the previous animal learned and generalize them and then remap them onto the new lifestyle. So now flying instead of crawling, and nectar instead of leaves. So the idea is that these memories, much like that bottleneck that happens during embryonic development, where the past generations give you genetic material, but then development is a creative process whose job it is to then reinflate that information into a new form and function. The same thing happens to learned information within the lifetime of a single animal. And so we can start to think about memories, and by memory I mean more broadly anything that carries information from past instances of you, so from your past self. So behavioral memories are messages from your past self, and genetic information are memories from the past version of you in your lineage. And in all of these cases, the current self, whether that be the cognitive being that is you, or the embryo that is trying to assemble itself from the single egg stage, does not actually have any access to what the past was like, right? And you're at any given moment, you don't have access to the past. What you have access to are the engrams, the memory traces that learning and past experience has left in your body. So So these are generalized and compressed representations of particular instances, and now it's your job, and as a cognitive system, we're continuously doing this and continuously refining our story about ourselves and the outside world by interpreting those memories. So it has this bottleneck feature, and anybody who does machine learning will recognize this kind of thing immediately, where you have a very thin bottleneck in the middle of something like a variational autoencoder or whatever, where what you're doing is you're doing some compression and you're throwing away some redundancies back here, but now here you have to reinflate that information, and it is precisely the fact that you have to reinflate it, whether that be the genetic information of a zygote, or whether that be the memory traces in a cognitive being trying to persist through time. It is that creative problem solving on this end that tries to adapt to the current situation given the information that it has, and all of that is described here. So the idea then, again, just to summarize, is that I think biology assumes from the start that the hardware is unreliable, which is very different from how we currently build computational systems, where we have a separation of layers, where you assume the layer below actually is going to do its job correctly. And biology realizes that environment and even your own parts are guaranteed to change, right? The one thing that's guaranteed is that there will be mutations and change. And so we can't overtrain on our evolutionary priors. And much like with cognitive systems, you care less about what your past self was doing with those memories, but you have to reinterpret them on the fly, right? It's the present, and the need to move into the future is what actually matters. So we started by trying to think about kind of generic connectionist ideas about, for example, the progressive generalization and recognition of layers in neural networks as different components of molecular gradients, and then the cells and tissues and body plans and so on. And now we have some very specific ideas that we're testing using the formalisms of NCAs, neural cellular automata, autoencoders, calculating things like causal emergence in cells that are not neural. So Tononi's lab and other people measure metrics of causal emergence like phi and so on in different kinds of brains under different conditions, to try and understand to what extent there is a mind, an integrated mind there looking outwards. And we can do all of that with the bioelectrics and the calcium imaging of the cells and the developmental systems that we work on. We are building models of genomes as generative models in development, diffusion models and so on. Lots of interesting conceptual frameworks available now, and we really welcome collaborations with all the people here and neuroscientists in general to help us take some of the tools that are being used to understand learning and information processing in artificial systems, but also in natural systems and in the brain, and various other things like active inference and perceptual multi-stability and so on. To try to apply them to decision-making in non-neural cells. And just to close here, I want to show you one example. I mean, I've made the claim that that biology has this sort of beginner's mind idea that the exact same hardware is perfectly willing to kind of confabulate new models of itself and the outside world, and do new things in novel circumstances, to make something adaptive. So I want to show you just one quick example of this. So here's a little life form. And if I asked you what this was, you might think that this is some sort of primitive organism that I got off the bottom of a pond somewhere. And you might also try to guess the genome, and you might think that the genome would be sort of indicative of some kind of primitive organism. And if you were to sequence this, what you would find is that it's 100% Homo sapiens. So these are adult human cells that have been given the opportunity to reboot their multicellularity. And what they end up doing is something we call making an anthrobot, because we think this is a biorobotics platform, and it makes these come from adult human patients donating tracheal epithelial cells. So it makes this little autonomous self-motile creature. They have some interesting properties. For example, if you seed a plate of neurons like this, so the red stuff is human neurons, we put a big scratch through it, so this is a wound, and you've killed off a bunch of neurons here. These anthrobots will gather in the wound. They make a group of them. We call it a superbot cluster of 10, 12 of these bots. And within about 4 days, if you lift them up, you see that what they're doing here is they're knitting the two sides of the wound together. So they have this ability to heal these neural wounds. Now, who would have thought that first of all, your tracheal epithelial cells that sit quietly in your airway would be able to make a self-motile little creature like that, and furthermore that it would have this kind of property. This is we're just scratching the surface here. This is the absolute first thing we look for, and we found this. I think they probably do a million other things that we're in the process of discovering. But the point is that these are genetically unmodified cells. These are perfectly normal cells from adults. That anthrobot looks nothing like any human embryonic stage, right? It doesn't look like a human embryo. There's never been any selection to be a good anthrobot. There's never been any anthrobots. So the form, the function, the capabilities of these anthrobots, and we're currently beginning to test them for memory, for learning, for preferences, for all kinds of other problem-solving skills. All of these things are novel portions of that latent space that surround the human genome, and the exact same hardware, the same human genome, is capable of being used to make lots of different kinds of creatures with different shapes, different behaviors, and it's precisely because of this problem-solving capacity, which in turn ultimately is driven by the unreliability of the biological hardware. So that ratchet leads directly from the unreliability of the biological material to a kind of very basal intelligence that permeates all of the different layers of life, leading up to having brains and nervous systems and so on. So because life is doing that, right? Because it is so interoperable, cells will grow in chimeras and hybrids with weird nanomaterials and scaffolds and electrodes. I mean, all of these things are completely viable. And because of that, and that's again because it does not make strong assumptions about what it needs to build, it's able to solve its problems in various ways. Because of that, what we're looking at here is an enormous space of possible bodies and minds. So almost every combination of evolved material, engineered material, and software is some kind of viable agent. So we have hybrids and cyborgs and every kind of combination, and some of these things already exist. Many of these things will exist in the coming decades, all sorts of different combinations. So on this space of the possible, of bodies and minds, all of Darwin's endless forms most beautiful, the natural variety of the evolved living world, is like a tiny corner, right? It's a tiny region of this space of possibilities. And this is all of these creatures are these forthcoming beings are an incredibly rich fodder for the neuroscience beyond neurons, to understand their emergent behaviors, their cognitive capacities, and of course, very, very importantly, for us to develop novel ways to relate to them ethically, because the old categories of life versus machine are no good. Well, they never were any good, but up until recently they were sort of good rough heuristics. But they're increasingly going to be irrelevant in the coming decades. And we really need to understand how to relate to beings that are nowhere on the tree of life with us, and to enter a kind of ethical synthbiosis with these various other beings, and thus gain a better understanding of what it is that makes us who we are, and what's special about being alive, and what's special about being humans, and so on. And so that brings me to the last thing, which I'll just say very briefly, because we don't really work on consciousness, and I don't usually say much about consciousness, but I think the one thing that we've learned from all this work, and you can see some of this in these papers, is that if you take these three things, any of these three things, as the kind of evidence that you use to solve the problem of other minds, right? So molecular mechanisms, evolutionary kinship,
or problem-solving behaviors. And so when you look out into the world and you deal with other humans or other animals, and you decide that they too have a mind and an inner perspective and consciousness, usually we use one or more of these criteria, right? It's either because of what they're made of, because they are related to us, and you know that you are conscious, so you assume that other things that are closely related to you might be as well. Or sometimes from behavior. Some people like to make those decisions based on their capabilities and their functional behaviors. If you take any of those things as important for detecting consciousness, then you really need to take very seriously the idea of other organs in the body that are not linguistic, that can't make a good verbal case for their own consciousness, and nevertheless have the same molecular mechanisms, the evolutionary closeness, and the problem-solving competencies of brains. Okay, and so that becomes very serious. And of course, I think it actually goes well beyond non-linguistic body organs, but in particular body structures. And that's because we've now shown that the bioelectrics that supports the relevant dynamics in the brain are actually extremely ancient and very widespread throughout the body. So here in this preprint, Nick Rouleau and I, we sort of go over all the currently popular theories of consciousness and ask, do they really say anything about brains, or are they much more generic than that? And actually, almost all of them apply to all of the tissues in your body. So I'll stop here and just summarize the following: that all of the mechanisms, meaning ion channels, gap junctions, neurotransmitters, all of that stuff that underlies neural activity are ancient. And they function in all cells in the body and across phylogeny, roughly from the time of bacterial biofilms. So the brain really developed its tricks and kind of sped up those processes, but they are very ancient and very widespread. The role of bioelectric networks as cognitive glue, as policies that allow integration of information across time and space and align the parts of a system towards higher level goals, is also very highly conserved. Our nervous systems are held together by bioelectrical signaling, and that's why we are more than just a pile of neurons, right? We are also collective intelligences, and that role for bioelectric networks is also extremely ancient. So not only the mechanisms, but also the algorithms and the policies are conserved. And for that reason, many of the tools of neuroscience, meaning both the techniques and the conceptual frameworks, are applicable broadly outside the brain, and doing that, applying them outside the brain, leads to novel discoveries. You can see here these are reviews of various aspects of regenerative medicine that are being enriched by taking the tools of neuroscience and in particular the understanding in neuroscience that there are multiple levels of explanation and of organization that are important, and applying them to other processes in the body to remind us that the molecular biology level is not a privileged level of causation. It is not the only level at which we can do useful interventions and so on. And I think that expanding neuroscience beyond nervous systems in that way enables really a very rich and novel roadmap to regenerative medicine. And in particular, this is because creative problem solving, learning, and so on are not brain specific. The sciences that study the self-assembly of bodies, meaning developmental biology and regenerative biology, and those that study the autopoiesis of minds from those ingredients, I think have a lot to teach each other. I think there's a lot of useful enrichment and information feeding back and forth from developmental biology to neuroscience and so on. And the broader implications of this connect with lots of issues around unconventional embodied minds. And so this is diverse intelligence research in terms of cyborgs, robotics, AI, and all other kinds of implementations that are doing some of the same things that life is doing in aligning parts towards a larger cognitive light cone and enabling an emergent mind that is a collective intelligence made of parts that obey the laws of physics. So more details on all of this can be found here. And most importantly, I need to thank the students and the postdocs who did the work. So all of the stories that I showed you today were done by these amazing people. You can see them here. We have lots of great collaborators. I want to thank our funders who have, at one point in time or another, have supported the work that I've shown you. The illustrations were done by Jeremy Gay, and again, disclosures. These are companies that are supporting some of our work and licensing some of the discoveries that have come out. And of course, most thanks of all to the biological systems that do all the heavy lifting and have taught us so much about what it is to be an embodied mind. Thank you so much.